Welcome everyone. In this video, we are going to learn about the general concept of genomic organization. Let's get started, shall we? Before we dive into our topic, we must know what is a genome. As we all know, every living cells contain a sequence of DNA. Now, all of this DNA contain cells, which is known as genome. One copy of genome can be found in nearly every living cells in our body. It contains all living organisms' genetic information. So, what is genomic organization? Genomic organization is the study of genome from its organism and its environment. Genomics examines all of the genes together, its interactions and effects.
Next is the organization of eukaryotic genome, a sequence of 4 nucleotides arranged in a certain pattern and coding information constitutes an organism's genetic material or DNA. The linear arrangement of DNA components and their partition into chromosomes is referred to as the genomic organization. Genomic organization can also refer to the arrangement of DNA sequences inside the nucleus and the three-dimensional structure of chromosomes. Eukaryotic genomes are the linear and follow the Watson-Crick double helix structural model. There are contained within chromosomes bundles of DNA and proteins, histone, known as nucleosomes. The protein coding genes in eukaryotic genomes are organized in exons and introns, which represent the coding sequence and intervening sequence, respectively, indicating the functionality of the RNA section of the genome. Eukaryotic genome organization. Basic facts on eukaryotic nuclear genomes are as follows. The first one is, eukaryotic genomes include two characteristics that pose a significant information processing problem. The standard multicellular eukaryotic cell has a substantially larger genome than a prokaryotic cell. Many genes can only be expressed in certain types of cells due to cell specialization. Second, a huge amount of DNA that doesn't direct the synthesis of RNA or protein is included in the reported 35,000 genes in the human genome. Third, the eukaryotic DNA is intricately organized. The DNA protein complex known as chromatin is not only linked to proteins, but is also structured at a higher structural level than the DNA protein complex in prokaryotes. And the last one is, eukaryotic cells have a significantly higher concentration of DNA in their nuclei than prokaryotic cells.
For the next one, we will talk about prokaryotic cell. Did you know prokaryotes means before nucleus? Chromosomal DNA in prokaryotes is present in structures called nucleoid. The bacterial chromosome is a single, circular, double-stranded DNA molecule mostly attached to the plasma membrane at one point. It does not contain any histone protein. Bacterial chromosomes are huge, but still it gets fitted inside the cell. This occurs because of supercoiling of DNA. If you guys ever wondering what is supercoiling of DNA, it is the process where prokaryotes compress their DNA into small pieces. In bacterial genome, the circular DNA is supercoiled because of addition of turns in the double helix structure of DNA. Last but not least, this is the picture of polycistronic transcript. What is it about? It is used to describe an mRNA corresponding to multiple genes whose expression is also controlled by a single promoter and a single terminator. Polycistronic mRNAs are also called operons.
Next is the genome in three-dimensional nucleus. Firstly is the nuclear lamina. In mammals, genome is contained within the cell nucleus. Genome organization is guided by context with several nuclear substructure, which is the nuclear envelope and its lamina. The outer nuclear membrane and inner nuclear membrane of nuclear envelope are populated by its transmembrane protein, which associate with the lamine proteins. Interaction between the lamina and chromatin regulate the position of chromosome and various other activities. Next is the nuclear pore complex that involved in regulating gene expression in chromatin organization. The nuclear pore complex mediates all transport between nucleus and cytoplasm. They are very large, in range mass from 60 million to 100 million Da. Nuclear pore complex perforate the two lipid bilayers of the nuclear envelope and composed of greater than 30 different nucleoporine proteins. And the third one is the nucleolus. Nucleolus are dense structures that are visible by light microscopy, where rRNA synthesis and pre-ribosome assembly occur.
Next is genome sequencing. Genome sequencing is about figuring out the order of DNA nucleotides or bases in a genome. For example, the order of A, C, G, and T that make up an organism DNA. The purpose of genome sequencing is to allow the microbiologist to link genetic characteristics of individual microbes.
How are the genome sequence determined? The genome sequence determined by a shotgun sequencing. A shotgun sequencing is randomly breaking up the genome into small DNA fragments that are sequenced individually. And then a computer program looks for overlaps in the DNA sequences, using them to reassemble the fragments in the correct order to reconstitute the genome. Why the shotgun method is used to determine the genome sequences? The shotgun method is used because it detects a large number of DNA polymorphism, and then the shotgun method is more complete and less artifactual coverage of the genome. Lastly, it improves the speed and cost of the genome sequences.
Chromosomes are made up of a DNA protein complex called chromatin that is organized into subunits called nucleosomes. The way in which eukaryotes compact and arrange their chromatin not only allows a large amount of DNA to fit in a small space, but it also helps gene regulation and control of gene expression programs.